So the next thing you're going to do is go to number 25, and for that we need to build glucose. Okay, glucose has the chemical or molecular formula of C6 H12O6, which is something you should memorize, and which I clearly have memorized. I really do. Just blanked out for a second. Okay. For this, I've already made the molecule. It's a little tedious to make, and it's pretty big compared with the other things. Okay. You have a diagram in your lab. Okay, and I want you to follow along with that. Okay, this is carbon number 1, 2, 3, 4, 5, 6. Okay, the numbering is actually really important because it lets us keep track of what's attached to each one of the carbons, and it makes a difference because, as you will see, the different monosaccharides, the 6 carbon monosaccharides, all have the same chemical formula, C6H12O6, but the way things are attached are different. Okay, you might see a lot of oxygens on here, right? Which hopefully you recognize that means there's a lot of polar bonds. And remember, glucose can make hydrogen bonds with water. Every time there's an oxygen, there's a partial negative, and the hydrogens attached to those oxygens have a partial positive. Okay, so what I want you to notice is that, so we have a lot of these hydroxyl groups, or these OH groups that I talked about before. Right, so we're going to see those all over the place. That's the functional group that is in this molecule. Okay, and then it's also circular, right? And just like cyclohexane, it has a flat structure that's more or less flat, circle structure in the middle, and then basically, if you think about it that way, then things are just poking up and down above or below the ring structure. Okay. And if you look on your diagram, you'll see that some of the OH groups are pointed upward and some are pointed downward, and that actually correlates to how they are set along on the ring. Okay, so here we have our oxygen. That means this is carbon number 1, and the OH group is pointed up, and in your drawing, it is drawn pointing up. That means it's above the ring. Okay, so first I attach the carbons, right? And hopefully you can see that carbon ring, okay? And then starting with carbon number 1, we have the OH group. So that's carbon number 1. Trying to figure out the best way to show you guys this. Carbon number 1 has an OH group going above the ring and a hydrogen going below the ring. Carbon number 2 has an OH group going below the ring and hydrogen going above the ring. Carbon number 3 has the OH group above the ring and the hydrogen below the ring. And that brings us to carbon number 4. Carbon number 4 has its OH group below the ring and its hydrogen above the ring. Okay, and then we have carbon number 5. Okay, this is carbon number 5 here. Okay, carbon number 1 and carbon number 5 flank the oxygen that's in the ring. Okay. So carbon number 5 has this whole thing up here. It's a CH2OH group above the ring, and then below the ring it has the hydrogen. Okay, this is carbon with 2 hydrogens and then an OH group, and you can just say CH2OH. Okay, so that is glucose. Okay. I also built galactose, which is glucose's very close cousin. It is also a 6 carbon sugar. It has a very similar structure. Okay, you can see that if you look at the structures in your activity, but we'll go through it as well. Okay, so same ring structure, same thing with the OHs either going above the ring or below the ring, depending on which one you look at, or below the ring here. Okay. So this here is carbon number 1, okay, and it has its OH group above the ring, just like glucose did. Carbon number 2 has its OH group going below the ring, just like glucose did. Carbon number 3 has its OH group going above the ring, also just like glucose. When we get to carbon number 4, though, this is where we have a difference. Okay? In glucose, carbon number 4 has its OH group going below the ring, and in galactose, it's above the ring. Okay? And then we get to carbon number 5, and that's exactly the same as in glucose. So the only difference in these two molecules, glucose and galactose, is when we look at carbon number 4 and whether or not the OH group is going above or below the ring. Okay, galactose has it above the ring, glucose has it below the ring. All right, so these are two monosaccharides, and they are used to make larger carbohydrates called disaccharides and polysaccharides. A disaccharide, di means two, so disaccharide is going to be two of these put together. Polysaccharide would be more. Okay, you need to follow along and look in your lab at the diagram of how galactose and glucose are linked together to make a disaccharide. Okay, so first of all, we need to make sure that we have galactose on the right side. Okay, galactose is the one that has the, so on carbon number, it's always fun to orient yourself here. Carbon number 4, that's right, this one. Carbon number 4 has its OH group above the ring on galactose. Okay, and carbon number 4 in glucose has its OH below the ring. So what I'm going to do in order to make lactose, we need to link these together, okay? And it's called a dehydration synthesis. Dehydration because water will be pulled out every time it happens. Synthesis because something is being made, okay? So the first step is to go to carbon number 4 on galactose, okay? And we're going to take off the whole OH group on galactose, okay? That leaves this carbon without a bond, okay? So, and that's where we're going to link it to glucose. With glucose, we're going to go to carbon number 1, okay? That's right next to the oxygen. And we are going to take just the hydrogen off the OH group, okay? We are not taking this hydrogen that's by itself. We are going to take the hydrogen off the OH group, okay? So I'm going to take that, okay? And then where this oxygen that used to be part of an OH group on the glucose doesn't have a bond, that's going to make a bond with the carbon there from the galactose, okay? This new bond, let me put this down for a second so I can make it, okay? This new bond right here, this is, I wish I could color it. This new bond right here is the bond that's holding these two monosaccharides together, and it has a special name, not because it's any different than any other type of covalent bond, it just is a covalent bond, but because it links the monosaccharides together, we call it a glycosidic linkage. Okay, there's lots of different types of glycosidic linkages, like 1,4 glycosidic linkages. We're not going to worry about that. We're just going to call it a glycosidic linkage. Okay, so monosaccharides are linked by glycosidic linkages in order to build bigger carbohydrates. If we had more chemistry kit, I could build another one of these and add another one on and another one on, and then you'd be building a polysaccharide like starch, for example. Okay, an important thing to remember is that, remember we pulled the OH group off of the galactose on carbon number 4 off the galactose, and the hydrogen off the OH group on carbon number 1 of glucose. And we're going to then put these together, and what molecule does it make? It makes water. Okay, so when you link two monosaccharides together through dehydration synthesis, a molecule of water comes out. That's why we call it dehydration synthesis. Okay, this is what we see for all of the macromolecules that are polymers. We put the monomers together by dehydration synthesis. So you see the same thing with nucleotides being linked together to make nucleic acids, and the same thing with amino acids being linked together to make proteins. Cytosine. The next thing you're going to do is look at nucleotides for nucleic acids, and we're on number 32. There is a diagram of both adenine and cytosine. We're just going to build one of them, and I've already built it. The molecules are really big, just the monomers are, and so they take a little bit of getting used to. One thing to notice is that you have a ring structure here, and that actually is a 5-carbon sugar that is incorporated within the structure of the nucleotide. Okay. There is, however, more to it than just a regular monosaccharide, right? The monosaccharide would not have this other stuff on it. So that's one way you can tell the difference. We don't have in our chemistry model kits, we don't have phosphorus, so we cannot make the phosphate group. That's with the P with the 3 or the 4 oxygens around it. So we're going to represent that just with purple, a purple ball. Okay, so that is our phosphate group. That makes things a little bit harder. We cannot actually make a dinucleotide using our kits because we really need a phosphate group. And phosphorus can make a lot of bonds. So we don't have anything that matches it. Okay, so let's orient this a little bit. This is the phosphate group. Here is the 5-carbon sugar. Okay, so the phosphate group is attached to carbon number 5. This is carbon number 1, 2, 3, 4, and 5. Okay, and when it's in a nucleotide, we say, instead of just 1, 2, 3, 4, 5, we say 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. And that reminds us that we're dealing with a nucleotide. Okay. And then we have this stuff up here, which is the base portion. Okay, so let's first look at the sugar. Okay, on carbon number 1, we have the base attached and a hydrogen. Okay. Carbon number 2, we just have 2 hydrogens. Okay, there's a difference between a DNA nucleotide and a RNA nucleotide, and that is right here at carbon number 2, whether or not there's an OH group or not on it. And if there isn't one, then it's DNA. If there is, then it's RNA. Carbon number 3 has an OH group on it. This OH group is actually really important because this is where the dehydration synthesis reaction takes place between this nucleotide, and if you had another one coming in, it would attach here. Okay. And then we have carbon number 4 here, and that's where the carbon number 5 is attached, and then the phosphate group. All nucleotides are going to have a phosphate group and a 5-carbon sugar, and then also something called the nitrogenous base. It's called nitrogenous because it has nitrogen in it. Okay, you can look at the structure between adenine and cytosine and see that everything except the base portion is the same, and then when it comes to the base portion, that's what's different. Okay, so this is the structure that makes this molecule cytosine. Adenine has a different structure. You can look at that and see that. Okay, this thing's pretty complex, has nitrogen, double bonded oxygens, another nitrogen, carbon with a double bond, it has another nitrogen up here, and then a double bond here. You certainly don't need to memorize this or anything like that, but it is nice to see this whole big structure that is a nucleotide. This, of course, is what our DNA is constructed out of, a bunch of these. So we're going to come back and look at these some more, and we'll study the base portion in more detail later on in the quarter. Okay, it's really important for you right now to be able to recognize the 3 parts of a nucleotide: the phosphate group, the sugar, and the base. And then our last models that we're going to look at today are amino acids. So amino acids are used for making proteins, and proteins are really important in the body, and my nerdy self thinks that they're the most fascinating macromolecules because they make proteins, and proteins are super complex and do all sorts of jobs in the body. So they're really, or in the cells, so they're really important. Okay, so we have serine and threonine, and I'm actually going to take off, I have made these already, but I'm going to take off the R groups for just a moment and show them to you. So if you look at the structure there in your handout, you should recognize that every amino acid has an amino group that is a nitrogen with 2 hydrogens, okay? And then there's a central carbon. The central carbon has a hydrogen attached to it, and then that is where the R group is attached, so we'll come back to that in a second. And then it has this big thing up here called the carboxyl group, or the carboxylic acid. Okay, so amino acid, that's how it gets its name. Amino for the amino group, acid for the carboxylic acid. The carboxylic acid has a double bonded oxygen and an OH group, and it's all one thing. We often see it together. Okay. So for my other amino acids that I made, it has the exact same thing for the baseline structure. Okay, amino group, central carbon, and then carboxyl group. And what's going to be different are the R groups, and the R groups, of course, going to attach to that central carbon, and they're going to be different. So serine has the R group of CH2OH. Okay, so here's my carbon, my 2 hydrogens, and then my OH group, and I'm just simply going to take that and attach that to my central carbon. Okay, so now it's there. Okay, so amino, central, carboxyl carbon, and then the R group is hanging off of that. Okay, so that's what's different. And we can do the same thing with threonine. Threonine has a bigger R group, and believe me, there are many bigger R groups. I just picked small ones because otherwise it'd be really confusing. This one has an R group of C, a carbon with a hydrogen, and an OH group, and then a CH3, which is a carbon with 3 hydrogens. Okay, so this group is going to attach to the central carbon of my other amino acid. Okay, and again, same thing, amino group, central carbon, carboxyl group, and then this R group hanging down, and that's the only thing that's different between these 2 amino acids now. Okay. And now we're going to show how these will get together through dehydration synthesis to form a dipeptide. It would just have a dipeptide, means it has 2 amino acids. There's also a polypeptide, which means it has many amino acids, and then there's a protein. Polypeptide basically means a pretty long stretch of amino acids that are not necessarily a functional protein. The way this happens is very exacting. On one of your amino acids, and it doesn't matter which, I have serine here, you're going to remove the OH group off of the carboxylic acid. Not this one down here, but off of the carboxyl carbon. Okay, so I'm going to take that off. Now that carbon needs to make a bond, and for that, I'm going to take off a hydrogen off of the amino group. Okay, and now that nitrogen, remember, we're not going to worry about that other hole, so that nitrogen wants to make a bond, and the place where it's going to make a bond is to that carbon, that carboxyl carbon. Okay, so then what we have is now we have 2 amino acids that are linked together. Each amino acid has an amino group, a central carbon with an R group attached when I'm showing up, when I'm showing facing down, and a carboxyl group. Okay, the carboxyl group, oh, oops, I had it the wrong way. There we go. Now this is the R group, and this is the R group, and we have what's called the backbone. Okay, so if you ignore the R groups for a moment, you'll see that you go amino, central carbon, carboxyl group, amino, central carbon, carboxyl group, and that's called the backbone of the protein. Okay. Definitely the R groups are important, and we'll talk a lot about those later when we get to protein structure, but the backbone is important too. Okay. And then the 2 amino acids are linked together through a specialized, well, it's really just a covalent bond with a specialized name. It's right here between the carboxyl carbon of one amino acid and the amino group of the other amino acids, and that's called a peptide bond. Okay, that was made through dehydration synthesis. Remember, we took an OH group off of the carboxyl group and a hydrogen off of amino group, and we make water, right? So we're going to pull water out of this. When we make that bond, we will pull water out, okay? And then we have our dipeptide.